Hello, welcome to Chapter 40, Water and Electrolyte Balance in Animals. By the end of this chapter, you should be able to describe the relationship between osmoregulation and excretion. You should be able to describe how cells, tissues, and organs in marine fishes remove salts from internal fluids, and describe how cells, tissues, and organs in freshwater fishes gain salts from the environment, explain how terrestrial insects maintain water and electrolyte balance, and explain how terrestrial vertebrates maintain water and electrolyte balance. And again, I should point out that we're only doing a portion of the total chapter, so feel free to read whatever you want, but only the material in this lecture will be on the exam. So, an electrolyte is a compound that dissociates into ions when dissolved in water. Okay? Put it in water, it becomes ions. It is essential that we maintain a correct electrolyte balance. So our cells need very particular concentrations of sodium, chloride, potassium, and calcium in order to work correctly. So this is a problem whether you live in freshwater, marine, kind of saltwater, or terrestrial environments. You still have to maintain water and electrolyte balance. All right, so electrolytes move through organisms by diffusion, right? So this is a passive process in which the movement of uncharged substances goes down their concentration gradients. So help me remember, what does it mean if something moves down its concentration gradient?
Oh yeah, it means it's when it goes from where there's more of something to where there's less of something.
Good job, Bird. All right, so that's what diffusion means. Okay, now water, so electrolytes move by diffusion, water moves through organisms by osmosis. So, as you remember from 1113, osmosis is the movement of water down its concentration gradient across a semi-permeable membrane. So, if you have solutes that are separated by a selectively permeable membrane, like a cell membrane, water is going to move from the side where there's lower osmolarity to the side where there's higher osmolarity, okay? So, osmoregulation is the process by which organisms control the concentration of water and solutes in their body. And if you aren't able to do this, if you aren't able to control water and solutes, you end up with osmotic stress. Okay, this is when you have abnormal concentrations of dissolved substances. So, as we talked about in lab, some animals are osmoconformers, and that means they don't osmoregulate. Instead, tissues and cells are isoosmotic. What's that mean?
It means they're the same concentration of ions inside and outside.
Yes. Okay, so electrolytes, the different kinds of ions, have the same concentration inside the cells as outside. All right, so freshwater fish are osmoregulators, right? And their cells are saltier on the inside than the fresh water that they live in, right? Because all of our cells have solute in them. So the water wants to go inside of their cells. So these cells tend to gain water by osmosis, okay? And so the place where they're gaining water tends to be through the gills because they're kind of sealed up everywhere else. But their gills are permeable, so they gain water by osmosis through the gills. The gill tissue has higher osmolarity than the fresh water, which has lower osmolarity. All right, they tend to lose electrolytes by diffusion. So it's saltier inside of their cells than outside of their cells. So the electrolytes want to diffuse outside of their cells, right? Into the water. So what they need to do is they need to hang on to their electrolytes, and they also need to gain more electrolytes by actively transporting in the electrolytes against their concentration gradient. So they actually have to use energy to pump in the electrolytes that they need, okay? All right, so marine fish are also osmoregulators, and they have the reverse problem to the freshwater fish. So they live in water that is saltier than inside of their cells. Okay, so the water wants to leave their cells by osmosis. They tend to lose water through their gills by osmosis. And they tend to gain electrolytes by diffusion. So the electrolytes want to go from outside in, the salt water where it's saltier to inside, right? Inside the fish. So they gain electrolytes by diffusion, and then they have to actively pump the electrolytes out through active transport so that they don't have too many electrolytes. Okay? All right, so that's the problem of marine fish. Terrestrial animals are also osmoregulators, and so they tend to lose water by evaporation from gas exchange and through just the surfaces of their body. And so water tends to be lost by evaporation, right? Whenever we breathe out, we tend to lose water vapor. And we gain water by drinking and eating, and we lose water also through urine and feces. In terms of electrolytes, the electrolytes aren't really, we just take them in by eating, and we lose them by peeing and pooping, but they're not evaporating anywhere. Okay, so electrolytes can move by passive transport, meaning diffusing along an electrochemical gradient, meaning they go from places where there's there's more electrolytes to where there's fewer electrolytes. And if they're moving by passive transport, no energy is required to do that. Electrolytes can also move by active transport, so they can be pushed against their concentration gradient. So you can either directly pump electrolytes from where there is fewer to where there's more electrolytes, or you can use active transport to create an electrochemical gradient, and then use the gradient to transport ions. So I'll show you that in a second. Water. Unlike electrolytes, cells will use pumps to transport ions, which creates an osmotic gradient, and then water will follow by osmosis, right? So pumps will put more salt somewhere, and then the water will follow the salt, okay? The water will follow the salt by osmosis. Often water can go into cells using aquaporins, so these are these molecules with a little tiny hole in the center, okay? And the water goes through the hole. There is no active transport of water itself across membranes, right? So you could actively transport ions, and then the water could follow the ions, but the water itself cannot get actively transported. All right. So all of our animal cells contain amino acids and nucleic acids, right? And so you need them to synthesize proteins and RNA and DNA. But then we have extra amino acids and nucleic acids, and they get broken down into ammonia, or NH3. And the problem with ammonia is that it's toxic to our cells, because if we have high concentrations of ammonia, our pH is raised. And remember, what happens if you have too high a pH?
Your enzymes get denatured.
Yeah. So we have to get rid of nitrogenous waste. So some animals can excrete nitrogenous waste directly as ammonia, or it has to get, in other animals, converted into other nitrogen-containing compounds to be excreted.
Where did you get those weird stickers?
I didn't make these up. If you go to everybodypees.org, it's a campaign on kidney health. So feel free to read more about your kidney and kidney health at everybodypees.org. Okay, so let's think about a bird, a mammal, and a fish.
I like to think about birds all the time.
Yes, I can understand that. For each group, what do you think is the most important thing? Either how much energy it costs to make the waste, or how much water is needed to make the waste?
Well, as a dog, it's pretty easy to get water, but if I was a fish, it would be even easier to get water.
Yeah, that's true. So fish have lots of water.
Yeah.
So do you think fish are more worried about how much water they need or how much energy they need?
They probably worry more about how much energy they need.
Yeah. What about a bird?
Oh, well, as a bird, I have to say, I can't drink very much water because I'm very small.
That's true. So the amount of water required is probably a problem.
Yeah.
So maybe the amount of water is a bigger problem than the energy.
Yeah.
What about the mammal?
I don't know, maybe if you live in a very dry place, you can't get enough water, but otherwise it's probably not that big a deal.
Yeah. Well, let's look and see. All right, so there are 3 forms of nitrogenous waste, right? And the production of each of the 3 kinds of nitrogenous waste has benefits and costs, okay? So the first kind of nitrogenous waste is ammonia, and that's what fish make. It's very soluble in water. It requires a lot of water to be able to excrete it, but it's very toxic, so you can't have it lingering in your body for a long time. So it tends to be primarily used in bony fish and aquatic invertebrates, and it's very cheap to make it. And so if the fish are in the water, they have all the water they need, and they can just pee all the time, so they don't have to keep it in their bodies, and they can excrete it in their urine or diffuse it across their gills.
Wait a minute, they're peeing out of their gills?
I mean, a little bit.
That's crazy.
I mean, yes. All right.
Hey, I know somebody who looks like that.
Is it you?
No?
All right. So for a mammal, right, the waste product that they usually use to get rid of nitrogenous waste is called urea. And it is medium soluble in water. It takes about a medium amount of water to excrete it, and it is medium toxic. So you can hang on to it in the body for a while. It takes a certain amount of water to get rid of it, and it's fairly dissolvable in water. So you tend to find this as the nitrogenous waste product in mammals, most adult amphibians, sharks, rays, and skates. So it's made in the liver, starting with ammonia or amino groups from amino acids. It can be very energetically expensive to make urea. And it is diffused, it is removed from the body in urine in mammals, and then across the gills in sharks.
Ew.
All right, the third form of nitrogenous waste is uric acid. It is not soluble in water. Hey, Bird, what happens if you poop on a car?
Oh, I love when that happens. It's like a real win for me.
Yeah? So how hard is it to get the poop off the car?
Oh, it's real hard. 'Cause, like, bird poop is, like, very sticky and dry.
Yeah. All right, so uric acid is not very soluble in water. It does not require very much water to excrete it, though. So a bird who is very, very small and lightweight doesn't have to drink a lot of water to be able to excrete nitrogenous waste. It's not very toxic. So a bird or a reptile, for example, so reptiles often don't go to the bathroom very often, so they can hold on to it in their body for a long time without hurting themselves. Also, terrestrial arthropods, like insects and spiders, also use uric acid to get rid of nitrogenous waste. So they start the synthesis with nucleic acids. It can be very energetically expensive to make uric acid. And they tend to excrete it with feces. So, for example, birds make kind of a white substance with their feces. They don't make a separate urine product.
Yeah.
Yeah.
Wait a minute, you don't pee? No, I mean, I just kind of make one thing. It's like white with like black chunks in it. Wow. Just wow.
So the type of nitrogenous waste that an animal makes is related to its evolutionary history and habitat, right? And so you can see that there are trade-offs between the energetic cost of making the particular kind of waste that it is and the benefits of conserving water. So if you make ammonia, it requires a lot of water, but it doesn't take very much energy. So it's very efficient for a fish to do it, but it wouldn't be at all efficient for a mammal or a bird to make that. Uric acid requires no water loss, but a lot of energy, right? So it is not very toxic, so you can hold on to it for a long time, but it's quite energetically expensive to make, so it wouldn't be very cost effective for a fish, especially, or even a mammal to make it. Okay, let's talk about Osmo Confirmation versus osmoregulation in marine fish. So sharks, rays, and skates are osmoconformers, but their blood does differ from seawater. So the shark's blood will contain low concentrations of ions, but high concentrations of urea. And the presence of the urea increases the blood's osmolarity, so it's nearly isoosmotic with seawater. And so as a result, even though it's different ions, so there's urea in the blood, and then there's kind of salt water outside of the shark, this causes very little water loss because there's the net concentration of solutes is the same inside the shark as outside of the shark, even though they're the different solutes from each other. Yeah, it would be energetically expensive to prevent the toxic effects of high urea concentrations. And so sharks will get rid of salt by excreting salt against their concentration gradient. They have an actual specialized gland called a rectal gland that they use to actively transport the salt out. Lots of animals have epithelial cells that transport sodium and chloride, and they have the same membrane proteins that you find in the shark rectal gland. So, for example, you have marine birds and reptiles that have access to salt water but not fresh water, and so they actually can survive by drinking salt water and then excreting salt by glands in their nostrils. So, if you've ever seen movies of like the Galapagos iguanas here, they will just sit there sneezing all of this salt out of their noses. Marine fish excrete salt from their gills, and there are mammals that can transport salt in their kidneys. All right, so in the gills of freshwater fish, epithelial cells have a blank solute concentration than the freshwater outside, thus the freshwater is blank to the fish's tissues.
Wait, what?
Okay, in the gills of a freshwater fish, epithelial cells have a blank solute concentration to the freshwater outside.
So it's freshwater fish?
Yeah.
So their cells are saltier than outside?
Yeah. So do they have a higher or lower solute concentration?
I guess higher.
Okay, so they have a higher solute concentration. So the fresh water is blank to the fish's tissues, hyperosmotic, isoosmotic, or hypoosmotic.
Hyposmotic.
Yes. Right? Yes. This one. Okay?
Yes.
Okay, so salmon and sea bass are used as model systems for really challenging osmoregulation. So both sea bass and salmon will move between saltwater and freshwater. And so when they're in saltwater, they have serious problems with losing water and taking on ions that they don't need. When they're in freshwater, they have problems with taking on water and losing ions, right? So these are very different osmotic stresses in the two environments. But the same fish have to be able to live in both places. So in these fish, their gill epithelia have chloride cells, and in the salt water, the chloride cells export salt, and in the fresh water, the chloride cells import salt. So here's when the bass is in seawater, right? So these are the gill cells. And I guess this is outside, and then this is inside the fish, okay? And this is an epithelial cell in the gills, and this is something called a sodium chloride potassium cotransporter. It is in the basolateral membrane. Do you remember what the basolateral membrane is? Yeah, that's like the bottom of the epithelial cell, like near the inside of the body. Yeah. Okay, so this is inside the body here. All right, so the bass is in seawater, right? Very salty in here, and it's less salty in here, right? And so the salts, basically they're at risk of taking on salts, and so they'll actively pump the sodium, chloride, and potassium out of the fish, right? And when the bass is in fresh water, they are able to flip the orientation of these transport proteins. So, okay, this is still the outside of the fish, and this is still the inside of the fish, but now they have the cotransporter on the apical membrane, on the outside of the epithelial cell, and it is actively pumping in the ions that the fish needs from the fresh water. So the ions are moving against their concentration gradient again to be pumped into the fish, okay? So the transport proteins are actually able to flip orientation between whether they are on the inside of the cell or whether they are on the outside of the cell. The transport protein is basically doing exactly the same job, right? But when it's over here, it's taking sodium chloride and potassium from inside of the fish and pushing it out into the seawater. And when it's moved over here, it's taking sodium chloride and potassium from the water and putting it into the fish. Okay, water and electrolyte balance can be very challenging for terrestrial insects, and so Often people like to study really extreme examples of organisms that have serious water and electrolyte problems. So one of those would be something like a desert locust or a flower beetle. So if you ever found a flower beetle in your bin of flour, it lives its entire life cycle in a completely dry environment, yet somehow manages to survive. So let's look at some adaptations insects have to allow this. So the insect's surface is covered with a waxy layer. So here's the epidermis, and then on the outside you have a layer of chitin and protein, and then wax on the outside. So this is the insect's cuticle. Because of this waxy layer, little water can escape through the epidermis into the environment, so they're able to hang on to water. If you remember, this picture is a little bit hard to see, but you can see here's the outline of like a grasshopper, and it's got this system of silvery tubes called the tracheal system. And the tracheal system are these narrow tubes that allow for respiration inside of a terrestrial insect. And then these tubes open in these little holes called spiracles to the outside world. And the spiracles can actually be closed when it's very dry outside to minimize water loss. Okay, so, yep, insects can cope with the desert through their cuticle covered with wax and also by closing spiracles. There's a second mechanism that insects cope with the desert, there's a second mechanism that insects use to cope with the desert. And this is by carefully regulating the amount of water and electrolytes they excrete. So they have a structure called Malpighian tubules, and these tubules can filter pre-urine from the hemolymph. So the kind of blood squishing around inside of an insect is called hemolymph. And the Malpighian tubules will filter a pre-urine, and then the hindgut will process and modify the pre-urine into a more concentrated urine. So by the time the urine is ready to be excreted, all of the water that can potentially be taken out of it has been. So this is kind of the outline of your grasshopper here. Here's its mouth, and then here's its gut, and then over here in the middle of its gut is a Malpighian tubule, which are like these little fingers here, okay? So when there's a shortage of water, or a shortage of electrolytes in water, and the insect's osmotically stressed, then the electrolytes and the water from the pre-urine, so the water gets kind of held off over here from the hemolymph, and the things that are needed get redigested into the body and then returned to the hemolymph, and then whatever is left over, so here we go, so the electrolytes and the water can be removed and put back into the body, and then only the final urine and feces, which has had everything removed from it, gets excreted as waste. So in a terrestrial insect, 80 to 95% of the water in the filtrate is reabsorbed, and that's what allows these insects to live in really dry environments like deserts or flower bins. So we get to talk about this a little bit in Lab 10, but in terrestrial vertebrates, osmoregulation occurs primarily in the kidney. And so this is a human, basically. So the renal artery, right here in red, brings blood that contains the nitrogenous waste into a kidney, and the renal vein will carry the clean blood back out, okay? And then the urine formed in the kidney is transported into the bladder for storage. If you look up closer, which we will in a minute, the kidney is made up of nephrons. So an individual nephron is the basic fundamental unit of the kidney, and this is where the action happens that causes the water and electrolyte balance. So this is a single nephron here, okay? And so we're showing kind of with and without the kind of circulatory system around the outside, okay? So the first step here is the structure here called the renal corpuscle, filters the blood, and it forms a filtrate, like a substance left over after it's filtered. And that consists of ions, nutrients, wastes, and water. So that's going this way, this way, this way. And oops, I missed it. Till it gets to the proximal tubule, this little twist here. And that reabsorbs more nutrients, ions, and water from the filtrate and into the blood. Okay. Then it goes down this low part here called the loop of Henle. This is the loop of Henle. It has a descending limb and an ascending limb. And this creates a strong osmotic gradient in the interstitial fluid surrounding the loop. So this is the interstitial fluid around the outside of the loop. And so it's able to pull away excess water, okay, out of the loop of Henle. And then the distal tubule reabsorbs ions and water and helps maintain water and electrolyte balance if necessary. And then you go up here into the distal, well, this part is, where's the distal tubule? Here to the top, round, to the distal tubule. And then finally, the collecting duct is the part that leads the urine out of the nephron and into the bladder, where it'll eventually be excreted. Okay, and yeah, there's separate blood vessels that serve each of the nephrons. All right, so what's the order that the filtrate passes through the kidney?
Oh no.
You don't remember?
I know the renal corpuscle is first.
Yeah.
I know the collecting duct is last.
Yeah.
I think the distal tubule was before the collecting duct.
Okay.
I think the proximal tubule was before the loop of Henle, and then the loop of Henle.
So what do you think?
I think it's renal corpuscle. Oh no. Now I'm backwards.
All right, so it's renal corpuscle, then what?
Proximal tubule.
Okay, then what?
Loop of Henle.
Loop of Henle, then what?
Distal tubule.
Yeah, then what?
Collecting duct.
Yep, that's the order. So in fish, amphibians, and non-avian reptiles, meaning reptiles that aren't birds, they don't have a loop of Henle, and so their kidneys don't have the ability to produce as concentrated a urine as you find in mammals. But if you're a fish or an amphibian, like, who cares, right? You do not need concentrated urine. You live in water, so that's not a big deal. But reptiles do have a problem with water, right? Water may be very hard for them to find, so they need to conserve water in their urine. So the reptiles will produce uric acid paste and excrete it with feces. So they'll make kind of a white paste instead of much of a liquid. And some reptiles have a bladder that collects urine and stores it before emptying it into the cloaca. And so when they are able to find any water, the reptiles will drink and drink and drink, and they'll fill up their bladder with this dilute urine, and then they'll hang on to it. So they actually will keep the water in their bladder, until they can get more water. And so this is a way of hydrating them. This is one reason why it's kind of a bad thing if you pick up a lizard or a snake and it kind of pees all over you. You might think, ew, gross, whatever, but the lizard or the snake may not have access to water, and so you may have kind of deprived that lizard or snake of the ability to be able to survive till the next time water is available. So public service announcement, go out there and appreciate them lizards and snakes, but don't pick them up. Hopefully you learned that different habitats pose different challenges with regard to maintaining water and electrolyte balance. So we talked about how animals control their concentrations of water and electrolytes, and we reviewed diffusion and osmosis and transport across membranes so that we can talk about how marine fishes, freshwater fishes, and terrestrial insects are able to control concentrations of water and electrolytes. And then we kind of took a deep dive into what terrestrial vertebrates do with their kidneys.